Hello students, welcome to the EPG Pathshala. My name is Dr. Shiv Kumar Sood. I am Senior Scientist in the Division of Animal Biochemistry at National Dairy Research Institute, commonly known as ICAR and DRI, an institute located in Karnal within Green State of Haryana near Delhi, the capital of India. Today, I am going to talk about the nucleic acid sequence analysis from the paper Biostatistics and Bioinformatics. Friends, a gene in a cell is defined as any stretch of continuous DNA sequence present on the chromosome in the cell, which in combination with RNA polymerase and involved protein transcription factors, if any, will yield one diffusible RNA product after transcription. That means a gene is a contiguous sequence on DNA present in chromosome, covers the actual transcribed DNA sequence as well as any additional DNA sequence controlling the transcription of actual transcribed DNA. This additional controlling DNA sequence may be upstream of the actual transcribed DNA or within the transcribed DNA or even downstream of this transcribed DNA sequence. Therefore, we can say that a gene consists of actual transcribed DNA and the sequence controlling the DNA sequence, if any, on the same DNA molecule. In prokaryotes, the genes having the actual transcribed DNA and additional sequence controlling the expression of transcribed DNA, they collectively are called an operon. Each operon may have one protein encoding DNA sequence known as CDS or open reading frame. The operon transcribing mRNA encoding more than one protein is called a polycystonic operon. Otherwise, it is called monocystonic operon. In this module, you will download a prokaryotic DNA sequence using a genome browser for the construction of an operon with the prediction of open reading frame encoding the protein product under the control of ribosome binding site, along with its transcription start and termination site, as well as the promoter sequence containing -10 and -35 sequence boxes, which are regulating its transcription. You will use this constructed operon for designing a pair of primers for PCR amplification of the DNA sequence for gene cloning and expression of the protein product through prediction of protein sequence in silico. Genome browsers are available for browsing nucleotide sequences of complete chromosomes and genomes for many organisms. The genes are arranged linearly on the chromosomes and can be browsed using a genome browser, such as the one available at University of California at Santa Cruz. The DNA sequence coding cellular function components such as polypeptide or transfer RNA or ribosomal RNA. These are the functional workers in the cell on the genomes and the chromosomes can be identified. That is, functional components such as polypeptide, transfer RNA, ribosomal RNA can be identified in the newly sequenced DNA using computer programs. These identified functional components are the starting points for the construction of complete genes to understand their biochemistry and molecular biology. Therefore, we'll use bioinformatics tools for computational construction of genes in newly sequenced DNA and genomes. in prokaryotes are organized as operons. We can use the DNA sequence for the construction of operon in prokaryotic DNA. The DNA can be from the laboratory or it can be downloaded from the DNA data bank. Downloading the DNA from the DNA database requires saving the DNA encompassing the complete operon. Then this downloaded sequence is to be analyzed for prediction of the open reading frames along with ribosome binding site, its transcription start site, termination signals, as well as elucidation of regulatory promoter boxes having 6 bases located at -10, that is upstream of the transcription start site and at -35 positions. These are commonly called -10 box and -35 box. -10 box is also known as data box. In prokaryotes, the genes are organized as either polycystonic or monocystonic operons. They differ just by the number of genes encoding proteins, that is the number of coding sequences. Polycystonic operons encode more than one proteins, whereas a monocystonic operon encode only one protein. In both the types of operons, the encoded proteins are under the control of a common regulatory DNA sequence called promoter, having 2 boxes with conserved bases, one located at -10, that is upstream of the transcription start site, called data box, and the second at 35 bases upstream of transcription start site and is called TT GACA box. First of all, let us use Microbial Genome Browser to download necessary prokaryotic DNA sequence to be used for the construction of an operon. In the present example, we'll use an operon from Enterococcus faecalis, which encodes 2 proteins. We already know that this is an operon, but we will be using computer programs to actually see that whether the computer programs are able to construct the operon or not. The first protein is 88 amino acid containing phosphocarrier protein HPR, and second is 575 amino acid containing protein enzyme 1. For construction of the full operon, download DNA sequence will include the coding sequence and 5' and 3' untranslated regions as well as promoter sequence. Visit UCSC Microbial Genome Browser and enter Enterococcus faecalis V583 to go to the browser. On the UCSC Enterococcus faecalis V583 Genome Browser, enter phosphocarrier protein HPR in the input text box and click Go button. In the ensuing page, find the gene EF0709 encoding phosphocarrier protein HPR. Note that the direction of the gene is forward as indicated by right arrows in cyan-colored gene bar marked by red arrow in this slide. This shows that the coding strand is plus strand and not the reverse strand. Click on this gene marked by red arrow in this slide. Click on the gene bar and in the ensuing page, you will notice position of the gene EF0709 on the chromosome is from 662,613 to 662,879. Note here that this position on the chromosome includes nucleotides encoding the protein only. Go to the previous page using browser's back button and click on the left column previous item button marked with 2 left arrows, and here in this slide marked with a black arrow. In this slide, on the ensuing page, click on black box containing text EF0708 marked by green arrow in this slide, and you will reach page showing position of the gene EF0708. Note that the position of this gene ends at 666442. Now click on the back button of your browser and then click on the two right arrows button at the page marked by a blue arrow in this slide to reach to the next gene. Next gene is immediately downstream of the phosphocarrier protein HPR in the same direction and encodes enzyme 1. Therefore, EF0709 and EF0710 belongs to same operon, they appears to belong to the same. Again click two right arrows button so that user reaches page showing EF0711. This gene is in the reverse strand of the chromosome. Therefore, this gene is not part of the operon encoding 0709 and EF0710. Click on the black box containing text EF0711 marked by yellow arrow in this slide, and you will reach page showing position from EF0711 starts at 664651. Therefore, the nucleotides from 662443, that is the end of the EF0708, to 664650, that is the start of the EF0711, encompasses the complete operon for these two proteins. Now click back button of your browser so that the gene EF0710 appear. When you reach at EF0710 gene bar, right click on the cyan colored gene bar to open context menu as shown in the slide. From the context menu, you select Get DNA for EF0710. In the ensuing window, enter chromosome position from 662443 to 664650. Since the gene is encoded on the plus strand, do not check the checkbox for reverse complement. However, in cases where the gene is encoded on reverse strand, then check the box for reverse complement. Now click Get DNA button. In the ensuing window, select all the text and copy and then paste in Notepad. Save the sequence using Save As dialog box with the name hpre1.fa with all files selected in Save As type box. In prokaryotes, any stretch of DNA sequence on its genome is defined as open reading frame, abbreviated as ORF. If it has a start codon AUG or GUG followed by other codons encoding several amino acids and finally ending with a stop codon UAA, UAG or UG. This ORF is called a protein coding sequence, abbreviated as CTS, if it is transcribed and translated in vivo. Computational CDS discovery in newly sequenced DNA is to predict protein coding ORFs, followed by the search for similar sequences in already known protein databases in other genomes. If the protein is found in some other genome, then it is most likely to be an actually expressed protein. Consequently, this newly sequenced DNA is annotated on the genome by the name of the protein found in that other genome. However, if the predicted protein is not found in any other genome, then this newly discovered DNA is annotated as hypothetical protein on the genome. For gene prediction, visit GeneMarks at the shown web address, upload the saved hpre1.fa file or simply paste FASTA sequence of the downloaded DNA. Then select Prokaryotic in sequence type and click on Start GeneMarks button on the ensuing page. Click on gms.out link to reach the result page. This prediction shows two genes, first from position 171 to 437 and second gene from 437 to 2164. There is no spacer DNA between two predicted genes. On the other hand, the last nucleotide in DNA at position 437, which is the last nucleotide A in the stop codon TAA of the gene EF0709, is the first nucleotide, that is the A of the start codon ATG at position 437. This prediction for two genes appears to be correct because the predicted gene length of 267 nucleotides will give 88 amino acid HPR protein, and predicted gene length of 1728 nucleotides will give 575 amino acids containing enzyme 1 protein, as it is already known in other species. But the same need to be cross-checked with the prediction of a promoter and two ribosome binding sites as well as transcription start site along with row independent transcription termination site for the construction of complete operon. Visit Software website as shown in the address, which lists several gene prediction software including gene finding in bacteria using FGenesB, bprom and other programs. bprom is a bacterial sigma 70 promoter prediction program with about 80% accuracy and specificity. It is best used in regions immediately upstream from the open reading frames. Since we do not have information about the sigma factor for the expression of the genes in question, therefore click on FGenesB to find operon and genes in bacteria. In the following page, paste the sequence of the constructed operon, select Enterococcus faecalis and click Process button. This prediction shows two genes, first from position 171 to 437 and second from 437 to 2164. This confirms the gene prediction of the two genes using GeneMarks. Visit neural network promoter prediction site to check if this DNA contains any promoter upstream of the predicted start site of the first coding sequence, that is upstream of the start codon ATG at position 171 on the DNA sequence. Check the radio button for prokaryote with the radio button for include reverse strand no checked and set minimum promoter score at 0.9 and paste the fastest sequence and click submit button. The ensuing page shows a list of the predicted promoters. Only 3 predicted promoters are shown in this slide. The enlarged base letter enclosed in the box in this slide shows start of transcription site predicted by this software. The start codon ATG is present at position 171 on the DNA sequence. Therefore, promoter number 2 appears to be correct promoter. A putative purine-rich ribosome binding site, that is GAA, GGA, GA, could be located 9 nucleotides upstream of the start codon ATG of the first open reading frame for the genome for the gene EF0709. This shows that the promoter for the operon starts at position 102 of the sequence. Therefore, the second promoter can be selected. One could predict and retrieve termination site in bacterial sequences. To predict row-independent transcription termination site, go to Arnold's transcription prediction site and upload FASTA file. Set the options for forward strand only and click Run button. It predicted a row-independent transcription stem loop structure highlighted in blue and red color. To confirm this stem loop site for transcription termination, visit WebGester at Indian Institute of Science, Bangalore, where the database of terminators in bacterial genomes is housed. Here one can search and browse using an alphabet. Click on E and then select Enterococcus faecalis from the list, followed by the click on the link in the line for Enterococcus faecalis. On the following page, click Genome View as highlighted by an arrow in the slide, a hyperlink on the right side as directed. Enter the genome position on the chromosome for DNA to be used for prediction. That is the position which were used for downloading raw DNA sequence. So in the start text box, enter 662443, and in the end text box, enter 664650 and click submit button. Now click on the stem loop button as indicated by an arrow in this slide so that you reach the stem loop row independent transcription termination site for this operon. This shows the stem loop row independent transcription termination site for this operon. This completes the construction of operon for EF0709 and EF0710 genes encoding histidine phosphocarrier protein HPR and enzyme E1 respectively. All the sites in the operon are shown here in this slide. These include transcription start site with -10 and -35 boxes, purine-rich ribosome binding site located 9 bases upstream of the start codon for HPR protein, followed by termination codon of enzyme 1, and finally the stem loop for transcription termination. The correctness of the constructed operon containing genes EF0709 encoding phosphocarrier protein HPR and gene EF0710 encoding enzyme 1 can be tested through the amplification of the complete operon for its cloning and transfer to a recipient strain lacking this operon. If the operon is functional in the recipient strain, then this operon is constructed correctly and it can be used for planning protein engineering experiments to improve the functional performance of this operon. The amplification of the complete operon can be achieved using PCR, that is polymerase chain reaction. PCR is an in vitro method, that is in the test tube, for repeated replication, therefore resulting in large number of copies of the specific DNA fragments. The specific DNA fragment may be very small as compared to the complete chromosome DNA molecule. This is similar to replication of only target gene, say only one gene out of several thousand genes present on the chromosome. In PCR, 2 oligonucleotide primers, each complementary to the extreme of the opposite strands of the DNA, separate by the region to be amplified, directs the synthesis of complementary strands towards each other to produce an exact copy of the DNA flanked by primers. Repeating the cycle of 3 independent steps carried out at their defined temperatures double the number. That is increasing the initial copy number of the target DNA in a geometrical fashion. Approximately m2 raised to the power n number of copies are produced, where we call M is the initial copy number of the target DNA, and N is the number of cycles, PCR cycles. Therefore, in PCR, several copies of the target desired gene are produced. Each copy is called amplicon of the gene. The steps involved are denaturation at 94 to 97 degrees centigrades, followed by annealing at 50 to 72 degrees centigrades by a thermostable, and third step involved the extension of annealed primers at about 72 degrees centigrade by the thermostable DNA polymerase such as Taq polymerase. Initial denaturation for 5 minutes at 94 to 97 and final extension at 72 degrees centigrade for 5 minutes is also included. Success of PCR is basically depending upon the specificity of the primers designed using the target DNA as template. The mistakes here during primer design will either result in no application or non-specific application. The polymerase chain reaction primers are designed following general rules summarized at a page available at EMBL as shown in the slide. Primers are generally in the range of 18 to 30 bases with similar GC content, generally more than 50%, so as having annealing temperatures from 50 to 60 degrees centigrades for increasing specificity. The primer pair have similar melting temperatures, that is TM, so that both primers anneal at the same temperature. The primers must have low complementarity to each other with minimal secondary structure, that is minimal self-complementarity, particularly in the 3' region to reduce primer-dimer formation. And most importantly, the primer must be highly specific to the target DNA with no cross-hybridization to non-target DNA sequences to avoid amplification of non-specific DNA. Primers are always designed with the help of a computer program. A number of programs are available from commercial software suppliers and are also available freely online over the internet. Visit Primer 3 Plus site and paste the sequence in the main tab text box. You can specify the target so that acceptable primer pair will flank this target DNA to be amplified using PCR. A target is the sequence in which you are specifically interested in your template DNA to be amplified. It might be a repeat size, for example, a CA repeat, or a single nucleotide polymorphism, SNP site. This will help in repeat and SNP analysis using PCR-based amplification and subsequent nucleotide sequencing in detecting the repeats and SNPs in template DNA from sample in hand. In the present case, however, we are interested in the amplification of complete operon containing the promoter, structural genes, and row-independent transcription termination site. The promoter DNA sequence starts at position 102 of the sequence and row-independent termination site ends at 2213 base position. Therefore, bases from 102 position to 2213 position must be the target to amplify. Therefore, select these residues from 101 position to 2214 position and click on Target button as indicated by an arrow in the slide. This will target the bases from the numbers specified to include in the amplicon. In this way, the complete operon can be targeted for amplification for further use in cloning and gene expression. In the General Settings tab, change the concentrations of the divalent cations and DNTPs to match your PCR buffer concentrations. Monovalent cations at say 50 millimolar and divalent cation 1.5 millimolar. In the Advanced Setting tab, check the checkbox named Use Product Size Input and Ignore Product Size Range at the bottom of the page, and then manually set the minimum product equal to 2112, optimum product size equal to 2180, and maximum product equal to 2200. Now click on the Pick Primers green button located on the top right side of the window. The results page will appear in the browser. The results page appears. Save the primer sequences and use these sequences for primer synthesis. However, before synthesizing the primers, check the specificity of the primers that this pair of primers will not amplify any non-specific DNA in the target genome. Check the specificity of primers using in silico PCR. In silico means an experiment using computer only, that is a virtual experiment. In silico PCR is used to predict that the designed primer pair will amplify only the target DNA and it will avoid any amplification of any non-specific DNA in the target genome. Visit insilico.ehu.es/PCR to check if the designed primer pair will be able to amplify only the target DNA and not any other stretch of DNA in the genome of Enterococcus faecalis V583. Select Enterococcus and click Next button. Paste the primer sequences in the provided text boxes and select Enterococcus faecalis and click Amplify button. The result shows that only one fragment of 2171 base pair DNA will be amplified. Therefore, the designed primers are specific and can be used for cloning. The real-time polymerase chain reaction, that is real-time PCR, is used to amplify DNA, and the number of copies of template DNA can also be calculated using real-time PCR. Conduct real-time PCR using designed primer pair with genomic DNA isolated from Enterococcus faecalis V583. The PCR amplification products are then resolved on an agarose gel, and the amplified DNA product with size of 2171 base pair fragment is visible as highlighted by marked arrow. This amplified DNA harboring the complete operon for genes EF0709 and EF0710 may now be cloned. After cloning, it can be transferred to a receiving strain which require these two proteins for their functioning. To translate HPR DNA to protein, in silico use Emboss Transect tool available at European Bioinformatics Institute. This tool translates the given DNA sequence to a protein. We know that the DNA sequence starts with ATG codon, so just paste in the HPR DNA in the text box and choose frame 1 and bacteria to click Submit button. This result shows the translation product of 88 amino acids encoding HPR protein. Translate is another tool for translation of a nucleotide sequence to a protein sequence using specific mitochondrial or nuclear translation tables. It is available at ExPASI, that is Expert Protein Analysis System server at Swiss Institute of Bioinformatics. Dear students, in this module, you learned downloading a genomic DNA sequence using a genome browser. The downloaded sequence was analyzed using online programs for prediction of two genes, ribosome binding site, transcription start site, row independent transcription stop site, that is stem loop structure, as well as -10 and -35 boxes of the promoter. The constructed operon was used for designing a primer pair for amplification of the complete operon using PCR, that is polymerase chain reaction cycle. The designed primers were tested first for their specificity using in silico PCR and were found to amplify this operon from the genomic DNA of Enterococcus faecalis and were not able to amplify any other DNA. Therefore, the amplified DNA can be used in protein engineering for improving functional performance of the two proteins in Enterococcus faecalis. In addition, this operon can be transferred to some other strain, some other bacteria which need these proteins for its functioning. Finally, open reading frame of HPR was translated in silico to get protein sequence of 88 amino acids so that this sequence could be used for identification of the protein using mass spectroscopy. Thanks for visiting EPG Pathshala.